Assalamu alaikum to all of you. I'm Umar Qureshi. I'm from the team Medimartt. Medimartt is a platform for the medical students. As you all know that we are regularly trying to share some short videos on different topics. So here we come with another video. This is a short video on the blood, and this is the fifth part of the blood series. Okay, and to watch our previous 4 parts, please watch it on our official YouTube channel. Those are uploaded on our YouTube channel, so you can check it out. And after watching that only you will understand this part. Okay, so please guys, please go and please check out those videos. And we are trying to make some short videos only. Okay, we are not making these videos lengthy. Okay, so please guys, please check it out and please watch this video. Please don't skip in between, because if you will skip you will not understand anything. Okay, and please guys, subscribe to our channel, please share it as much as possible, and please do comment what topics like you need. Okay, so we will try our best to make the videos on that particular topics. So let's start. So in our previous video we have discussed some factors controlling or required for the erythropoiesis, and those were some general factors, then the hemoglobinization factors, then the maturation factors, then intrinsic factors of Kessel and all type of things. Okay, in our previous video we discussed this mechanism. We will discuss this mechanism again. So in when you feel the lack of oxygen in your body, which is also called as the hypoxia, this hypoxia stimulates the kidney, and this kidney basically releases the renal erythropoietic factor. Okay, and this renal erythropoietic factor this helps in the erythropoiesis. Okay, and this alpha globulin basically converts into the erythropoietin with the help of this renal erythropoietic factor. Okay, then these are some cells, stem cells, then this erythropoietin, and this goes into the stem cell, then the proerythroblast, then increase the RBC number. Okay, then it increases the hemoglobin concentration also, because when RBC increases, hemoglobin concentration automatically increases. Okay, and this hemoglobin increases, increased hemoglobin goes into the tissue organ, and this is a reversible negative feedback mechanism from tissue organ to the kidney. Okay, the negative feedback mechanism occurs. What is negative feedback mechanism? We will explain this in our general physiology videos, inshallah. Okay, so please stay tuned for that, and please watch our videos regularly, and please subscribe to our channel so that you get notifications of our recent videos. Okay, so let's start by new topics. So basically it is about the shape of RBC and advantages of this shape. Okay, so basically shape, the normal red blood cells are circular. It is circular and biconcave disc shaped. Okay, so this is the basic shape of the RBC. Please learn it by heart. Okay, now what are the importance or the advantages of the biconcave shape? Okay, so basically the thickness of RBC in its central part is not great, so that oxygen does not have to travel a great distance for the diffusion. Okay, so basically as we saw that the thickness of this RBC a cell, okay, this cell is basically in center it is thin and in periphery it is thick. Okay, so the thickness of RBC in central part is not great. The thickness is not great in the central part, so that the oxygen basically does not have to travel a great distance for the diffusion. Okay, so that is the point. Oxygen does not have to travel a great distance for the diffusion. That's the thing. Now because of the biconcavity, okay, surface area of RBC, so that the oxygen get bigger area for diffusion. Okay, so because of that biconcavity, as it is biconcave, okay, so this surface area of RBC also depends on that, then the oxygen basically get bigger area of diffusion. Okay, so that's the point. These are the things that are not very much important. Just remember this thing right now. Okay, now large surface area is provided for the absorption or removal of different substances. Okay, as due to the presence of the biconcavity, okay, the surface area increases. Okay, so due to this large surface area, So this large surface area is helpful for the absorption or the removal of different substances, like many substances can enter. Okay, so those substances are removed with the help of this large surface area. Now while passing through minute capillaries, can squeeze very easily. Okay, so if these red blood cells are passing through the minute capillaries, so these can pass through those minute capillaries very easily. That is the point. Okay, now moving on towards the next slide. And this advantages of absence of nucleus in RBC. As we all know that nucleus is absent in the red blood cells. So what are the advantages of the absence of that nucleus in the RBC? So basically it provides some extra space for the storage of large amount of hemoglobin within the RBC. As we all know that if something is absent, then we can get some space in that particular place. Okay, so that's the point, that the nucleus, when the nucleus is absent, so as we all know that hemoglobin is present in the RBC, so hemoglobin is getting a large or the extra space for the storage of that particular hemoglobin in that red blood cells. Okay, so at their maximum limit to facilitate gaseous exchange. So basically this what I want to tell you is that this absence of nucleus in the red blood cells is basically helpful for the storage of large amount of hemoglobin within the RBC. Okay, and the maximum amount or maximum limit is basically stored how much this RBC can basically acquire. Okay, so according to that, maximum limit is stored in that particular red blood cell, or a maximum limit of the hemoglobin is particularly stored. Okay, why it is stored and why it is helpful? So basically it helps in the a gaseous exchange. Okay, so from the carbon oxygen to carbon dioxide, that's the gaseous exchange. Okay, so that's the point. Now it provides special ability to change its shape when helps passing through the narrow capillary. When as we know that when the nucleus will be absent, so it can change its shape, which helps the passing through the narrow capillaries also. Okay, so that's the point. Now abnormal forms of RBC. What are the abnormal forms of RBC? Abnormal forms of RBC are microcyte, macrocyte, hypochromic, anisocytosis, polycythemia, erythrocytosis. There are more, but first of all we will discuss these 5. Okay, so first of all, what is microcyte? So when the RBCs are smaller than the normal in size, they are called as the microcyte. The normal size of the RBC, when it will be smaller than the normal size, it will be called as the microcyte. Now macrocyte, macro means big, micro means small. Okay, so that's why micro, all the answers are in its names also, in the names also only. Okay, so you can check it out. So microcyte, basically when the RBCs are smaller than the normal size. Then macrocyte, macro means big, so when the RBCs are larger than the normal in size, so they are called the macrocyte. Now what is hypochromic? Hypo means less, and or chromic means the color. Okay, so when the concentration of hemoglobin per RBC is less than the normal, the RBCs are said to be hypochromic. As we know that this hemoglobin provides the red blood cells the red color or the eosinophilic stain. Okay, so when this right now the RBC is properly stained and properly well colored, well red colored, so that's why it is called as the normal RBC. Okay, but when this hemoglobin concentration becomes less, okay, so the color of that particular RBC will also become less. Okay, so according to that, when the concentration of hemoglobin per red blood cell is less than the normal, the RBCs are said to be hypochromic. Okay, so that was that. Now anisocytosis, a variation in size of RBC from normal size is called anisocytosis. Means the size of that particular RBC is something like that. Okay, so if we can see the variation in the size of that particular RBC, then the normal size is called as the anisocytosis. Okay, now polycythemia or erythrocytosis. Okay, So what does that mean? Abnormal increase in red blood cell count. Okay, so cythemia means for polycythemia, it basically means the abnormal increase in the red blood cell count. Okay, poly means like increasing, or many you can say, so it is according to that. Now moving on towards the next slide. These were 5. Then some important terms you have to remember, like normocyte. What is normocyte? It is in the name in itself, like normo means normal. Okay, so RBC when normal in size is called as the normocyte. Now what is normochromic? Same, normal means, normo means normal. Okay, so when the concentration of hemoglobin per RBC is normal, the RBCs are called as the normochromic. So that was the normochromic. Okay, now hemopoiesis. What is hemopoiesis? So basically the process of formation of RBC under normal physiological condition is called as hemopoiesis. Okay, so that's the point about the hemopoiesis. This these are the important terms or important definitions we have to remember, very frequently asked in the items and the professional examinations. So please remember it very clearly. Okay, now hemolysis. Lysis means the breakdown, and heme means the breakdown of RBC, you can say. Okay, so the process of breakdown of RBC is called as the hemolysis. Now moving on towards some more things like peculiarities of RBC. What are some peculiarities of RBC? Peculiarities means what are some uniqueness of particular RBC. So as we know that there is no nucleus, so this will be a peculiarity of RBC, that is the absence of nucleus. Then as you know that there are no mitochondria and the glycogen, so that is also a peculiarity, absence of mitochondria and glycogen. Then there is no DNA and RNA, so absence of DNA and RNA. So synthesis of protein in RBC is not possible. Okay, so because of this DNA and RNA absence, because of this absence of DNA and RNA, synthesis of protein in RBC is not possible, and it contains hemoglobin. That is the most important point, so please remember that. Now moving on towards the next topic, that is the hemoglobin. Okay, so hemoglobin, what does this mean? Okay, so hemoglobin, as I already told you, that it is basically subdivided into the two parts, that is the heme and the globin portion. So that's the definition basically. So hemoglobin is a chromoprotein consisting of protein globin united with the pigment heme. Okay, so heme is the pigment and globin is the protein. Okay, so according to that, hemoglobin is basically a chromoprotein. Chromo means color and protein means you already know about the protein. Okay, so hemoglobin is a chromoprotein consisting of protein, that is the globin, and it is united with the pigment, that is the heme. Okay, so heme is basically united with the globin and forms the hemoglobin. Now the normal values of the hemoglobin in male, female at birth and at the first year. Okay, so according to that, in male, that is the hemoglobin, so range is around the 14 to 18 gram per deciliter, and average is around 15.5 gram per deciliter. Please remember this normal value very because this is very important, so please remember it by heart. Okay, and now the female normal value of hemoglobin is when you talk about the range, so it is around 12 to 15.5 gram per deciliter, and average is around 14 gram per deciliter. Okay, now at birth there is no range, directly we will talk about the average of the hemoglobin at birth, so it is around 23 gram per deciliter, and at the first year it is around 12 gram per deciliter. Okay, now let's move towards the functions of the hemoglobin. So functions of the hemoglobin are basically somewhat 4 functions mainly. Okay, so what what are the functions of the hemoglobin? So basically it transports the oxygen from the lungs to the tissue. As we all know that respiration occurs, so this is the or this helps in the respiration, like that transports the oxygen from the lungs to the tissue and carbon dioxide from the tissue to the lungs. Okay, as we all know that diffusion of gases and occurs with the help of this red blood cell and with the help of this hemoglobin. Okay, so this hemoglobin basically helps in the transport of oxygen from the lungs to the tissues. Okay, and carbon dioxide from the tissues to the lungs. Now it is an important blood buffer and maintains the acid base balance of the food. Okay, so basically for this point the blood buffer, or what is buffer and what is the acid base balance. Okay, so please refer to our biochemistry series videos. Okay, and those are uploaded on our official YouTube channel, so please go and please check it out. Just now, right now in this physiology video, just remember this thing, that is it is an important blood buffer, okay, and it maintains the acid base balance of the food. Okay, so that's the point. Now formation of various pigments. Okay, now what are the formation of various pigments, like what pigments it helps to form? Okay, so basically bilirubin, stercobilin, urobilin, these are some important or various pigments that are formed with the help of the hemoglobin. Okay, now it reserves iron and protein. Okay, so basically it is the like it acts as the reservoir of the iron and protein. Okay, so that's the point. Now let's discuss this most important topic of this RBC, that is the fate of RBC or hemoglobin, very frequently asked in the items, professional examinations like written, vivas, both. Okay, so please remember this thing very frequently. Okay, so first of all, the destruction of the fragile RBC by reticuloendothelial system. Okay, so if the RBC is fragile, so this this will be destructed by the help of reticuloendothelial cells. Okay, now the release of hemoglobin occurs when the RBC is destructed. As you know that hemoglobin is present in the RBC, so when it is destructed, so hemoglobin will be released. Okay, now hemoglobin is phagocytosed by the macrophage and is splitted into. Okay, now at this point hemoglobin will be phagocytosed by the macrophages. Like what are the function of macrophages? The function of macrophages is like a phagocytosed or phagocytosing. Okay, so that's the thing. Now hemoglobin is phagocytosed by the macrophage and is splitted into the two main parts, that is the heme part and that is the globin part. Okay, so here the hemoglobin will be like subdivided into the two parts, that is the heme part and the globin part. Okay, now this some part of the globin is basically converted into the amino acids and it is converted into the blood. Okay, or it is basically dissolved into the blood. This amino acid is dissolved into the blood. Now this heme part is basically, heme is broken into the biliverdin. Okay, this heme part is basically divided into the biliverdin plus iron. Okay, then this iron is either stored or is either reused. Okay, so that's the thing. Now biliverdin, this biliverdin is converted into the bilirubin. Okay, now this bilirubin is basically combines with the albumin. Okay, and this bilirubin combines with the albumin and which are present in the plasma. Okay, so this bilirubin combines with the albumin and forms the free bilirubin in the plasma. Okay, now this free bilirubin, it is enters into the hepatocytes, that is also called as the liver. Hepatocytes are the cells of the liver. Okay, so basically bilirubin enters the hepatocytes in the liver. Now the glucuronic acid combines with the bilirubin by the action of the glucuronyl transferase. Okay, why it is happening? Because bilirubin is converting into the bilirubin glucuronide, which is also called as the conjugated bilirubin. So what type of thing is helping to convert the bilirubin into the bilirubin glucuronide, and that is due to the glucuronic acid, and this glucuronic acid basically combines with the bilirubin, and these two when these two combine, the bilirubin glucuronide is formed by the help of or by the action of glucuronyl transferase. Glucuronyl transferase is the enzyme which is helping to convert into the bilirubin glucuronide when this glucuronic acid and the bilirubin is combining. Okay, so bilirubin glucuronide is formed and conjugated bilirubin. Okay, now this bilirubin glucuronide or conjugated bilirubin is excreted into the intestine with bile. Okay, with the help of bile, this bilirubin glucuronide is excreted into the intestine. Now bilirubin glucuronide in the intestine, basically it converted into the stercobilinogen. Okay, with the help of the bacterial action in the large intestine. As we know that some bacteria are present in the large intestine, so with the help of this bacterial action, this bilirubin glucuronide, which is excreted into the intestine, this is converted into the stercobilinogen. Okay, then this stercobilinogen, some amount of stercobilinogen with the help of blood or via blood, basically goes to the kidney, where it is known as the urobilinogen. Okay, so what is happening here? The stercobilinogen, which was converted from the bilirubin glucuronide with the help of some bacterial actions in the intestine. This stercobilinogen is basically goes to the kidney with the help of the blood. Okay, where it is converting into the, or where it is known as the urobilinogen. In the kidney it is called as the urobilinogen, and by which medium it is going? It is going with the help of the blood. Okay, now this urobilinogen is basically converted into the this urobilinogen in the kidney basically converted into the urobilin, which excreted with the help of the urine, and through which thing, or through which process this urobilinogen in the kidney is converted into the urobilin, and with and that is due to the oxidation process. Okay, so with the help of the oxidation process, this urobilinogen in the kidney is converting into the urobilin, and this urobilin is excreted basically with the help of the urine. Okay, and this stercobilinogen, as we saw that stercobilinogen, some amount of stercobilinogen goes with the help of the blood into the kidney, and some amount of stercobilinogen with the help of the stool it will be excreted out, but for that it should be converted into the stercobilin. So with the help of the oxidation process, this stercobilinogen is converting into the stercobilin, and this stercobilin is basically excreted out with the help of this stool. Okay, so these were the points about that. Okay, so this fate of the RBC is very important. Please remember it by heart. And guys, if you love the video, then please do share it as much as possible. Please subscribe to our channel, and please comment down if you don't understand anything, and we will make another video on that particular topics. Thanks a lot.